Hello everyone and welcome back to the Burlington Central Brain Bee Club YouTube channel. My name is Josephine and in this video we are going to go through chapter 2 of the Brain Facts textbook, which is focusing on senses and perception. So the previous video covered chapter 1 of the Brain Facts textbook, which is simply brain basics. So looking at basic brain anatomy, a couple of important things about brain waves, neurons, glial cells, and so on. So if you need that refresher, please go and check out that video. But without further ado, let's jump into the three main topics of the video, which are vision, hearing, as well as touch and pain. All right, so starting off with vision, we have an important diagram here to have a solid understanding of the anatomy of the eye. So the very outer layer of the eye is called the cornea, and it kind of gives your eye that glossy, watery appearance. It deals with protection and refraction of light, so it does help with focusing light a little bit into your lens, as well as protecting the eye from debris. We also have the iris, which is actually a muscle. It's a circular muscle that is able to contract or expand to allow a certain amount of light to enter through the pupil, which is simply an opening in the eye. If your pupils are dilated, more light will enter. If they are constricted, less light will enter. Situations where your pupil would be dilated include a fight or flight response. You would want to have more light entering your eye so that you can see very well and you can kind of resolve the situation a bit better because you can see very well. So the lens is a muscle as well that can thin and thicken in order to bend light or to refract light onto the retina. So it's a super important part. This is your eye's natural prescription, basically. And then the retina itself is found at the back of the eye, and it deals with light detection. This is where the light is actually being kind of put together and sent off to the primary visual cortex via the optic nerve. It's one of the 12 cranial nerves. It's the second cranial nerve, and it is essentially a bundle of ganglion axons that leave through the back of the eye. This spot that they leave is called the blind spot because there are no photoreceptors in the retina at this position. So let's first of all explain what photoreceptors are. They are essentially neurons that are able to process various forms of light. So we have two main types of photoreceptors. We have cones and we have rods. Cones deal with color vision, rods deal with black and white, so basically vision in the darkness. And your cones, there are three main types of colors that they process: red, green, and blue. So you might be wondering, how is it that we only have these three colored cones, but we can see an unlimited amount of colors? And that's because of the pattern of their activation. So for example, if you have a lot of red and blue cones activated, you would probably see a more purple color. So it really just depends on the level and the order of activation of these photoreceptors. And then rods, on the other hand, are really only active in the darkness. Interestingly, though, 95% of all photoreceptors are rods, which just goes to show how much power it takes to be able to see in the darkness. It's a pretty challenging thing for the eye. Perfect. So this is an important diagram, just kind of going over the anatomy of the eye and then the retina as well. But moving forward, we have another important diagram for vision, and this is kind of tracking how visual information is transported and processed. So it is broken up into the left visual and the right visual fields. This is just the kind of what you can see. So the left visual field is actually processed by your left eye. The right visual field is processed by your right eye for now. But notice that the retina of each eye is broken up into two parts. The inner part of the retina is called the nasal retina because it is closest to your nose, and the outer part is called the temporal retina because it's closest to your temples. So just remember that terminology. Now, the interesting part is that it kind of flips. So information from your, what we would call temporal visual field, because it's towards your temples, would turn out to be processed at your nasal retina. And then information from your nasal visual field will be processed by your temporal retina. So leaving the eye, we have the optic nerve, and then we have the optic chiasm, which is where the nasal pathways, so the one closest to the nose, this red one and this blue one here, they cross over. So remember how I mentioned we have something called a blind spot, which is where your optic nerve leaves the back of the eye because there are no photoreceptors there. But the nasal retina is able to cross over at the optic chiasm, and the left eye fills in the blind spot of the right eye, and the right eye fills in the blind spot of the left eye. So that way we don't actually see any blind spot in our vision unless we cover one of the eyes. And notice that the temporal retina always stays on the same side, whereas the nasal retinas are the ones that cross over. So now your right eye, some of its vision is on the left side, and so on. Then we reach the lateral geniculate nucleus and the pretectal nuclei. And the lateral geniculate nucleus is where the preganglionic neuron, so just basically this optic nerve and optic tract right here, hands information over to the postganglionic neurons, which we tend to call optic radiations. The pretectal nucleus just provides some information on visual reflexes. And then that information reaches the primary visual cortex, which is where visual information is now going to be processed. Now, another thing I actually forgot to mention is that the retina receives your visual information upside down. And so the primary visual cortex does a great job of flipping that back right side up. So moving on here, we have a couple of key points from that chapter about vision. The center of the retina is called the fovea, and it has high visual acuity. It has a high density of photoreceptors. The macula is a ring kind of around the fovea. It's important for reading and driving. And I think it only has green and red photoreceptors. It has the highest visual acuity, and when the elderly start to lose their vision, it's typically due to macular degeneration of that particular spot, which is why it's impacting reading and driving the most. There are 3 types of neurons in the retina. We focused on photoreceptors, but there are also interneurons and ganglion cells. Ganglion cells are the ones that leave through the back of the eye and form the optic chiasm. Interneurons are kind of in between those neurons. And then we have 2 visual processing streams. We have the dorsal visual processing stream, which sends information up to the parietal lobe to decipher the where and how of what you're seeing. It's basically allowing you to come up with an action plan for kind of how you want to move forward now that you've been able to identify what you're seeing. But you identify what you're seeing and you recognize it through the ventral visual processing pathway, which is sent down to the temporal lobe and again deciphers what you're seeing. It's able to recognize that. So that brings us to the end of the vision portion of this video. Next up, we have hearing. So we're just going to break down the anatomy of the ear and kind of quickly talk about what each part does. So from left to right, or from outside to inside, we have the auricle, which is simply a cartilage structure. It's what we see and what we call the ear. And it is simply just funneling air and sound waves into your ear canal, which continues to funnel that sound through to your tympanic membrane. So the ear canal is what's considered the outer ear. The tympanic membrane, now that it's been hit by sound waves, it vibrates and it causes the three bones of the middle ear, so the malleus, incus, and stapes, to vibrate as well. The malleus is also known as the hammer, the incus is also known as the anvil, and the stapes is also known as the stirrup. These are the smallest bones in the body. Now, the stapes is closest to the cochlea, this purple snail-like structure, and it connects to the cochlea via the oval window. Now, when the stapes vibrates, it causes pressure change in the cochlea, which generates vibrations in the fluid of the cochlea. So the cochlea has fluid in it, and the pressure changes cause waves. So when the stapes vibrates, it attaches to the cochlea via the oval window, and the cochlea is this purple snail-like structure that contains fluid. So when the stapes vibrates the cochlea, it causes the fluid itself to also vibrate, which the cochlea is lined by a basilar membrane, and the basilar membrane contains stereocilia, which are these hair-like cells that when they're moved, they cause an ion influx. And so all of this kind of vibration leads to the activation of the cochlear nerve, which sends auditory information to the auditory cortex. So really, what you need to understand in terms of the concept of hearing is that it is all based on sound and pressure waves. So it's basically just due to a kind of movement of air that causes sound. Now, these semicircular tubes kind of coming off of the cochlea have a slightly different function, but they're also very important. This is where balance and coordination is coming into play. You've probably heard that the ear is the center for balance, but this is why. So the semicircular tubes, there are three of them. One is in the y-axis, one is in the x-axis, and one is in the z-axis. Now, the reason being, it allows you to orient yourself and be able to recognize that, oh, I'm standing upright, or I'm lying down, or I'm upside down, right? It can also help with localizing sound. So being able to identify if a sound is coming from above you, below you, from your height. And all this information is sent via the vestibular nerve to the cerebellum, which also deals with balance. The cochlea's fluid-filled interior is also important for balance. So if you've been spinning in a circle for a long time and then you abruptly stop, you'll feel like you're still spinning because the fluid in your cochlea is still spinning. So just some interesting physiology here when it comes to the ear. And two important parts of the brain that deal with hearing are Broca's area and Wernicke's area. So Broca's area deals with speed production. It's in the frontal lobe. This is how you are able to articulate what you are thinking. Wernicke's area deals with speech comprehension. It's in the left temporal lobe, and this is how you are able to comprehend what other people are saying and how you are able to come up with comprehensible words. So those are two key parts of the brain that deal with hearing and speech production as well. Finally, we have touch and pain. And this is a diagram indicating where each body part is controlled by the precentral gyrus, or the primary motor cortex. So this is called the motor homunculus. So you can kind of get a gist of which parts of the brain process which parts of your body, which is very interesting. So this is kind of the pathway of sending pain signals. So the nerves that deal with pain processing are called nociceptors. And when you have a painful stimulus, also known as a noxious stimulus, it's going to travel via a nociceptor, which is a peripheral nerve, right? It's not part of the central nervous system because it's coming from a different part of your body. But there are two fibers that it can choose to travel by. One is a myelinated A delta fiber. One is an unmyelinated C fiber. Myelinated A delta fibers, because they are myelinated, they are able to send information much faster. Unmyelinated C fibers are unmyelinated. They don't have insulation from myelin, so they send information a lot slower. If a pain signal is transported via a myelinated A delta fiber, that means it's likely acute, sharp pain, right? It's something that you need to make your brain aware of very quickly. It could be burning yourself, it could be stepping on something sharp. And then an unmyelinated C fiber would tend to transport more diffuse kind of aching pain, so something that's more long-term, less sharp, maybe a more diffuse, so you can't really spot the specific location. Whereas, for example, if you get burnt, you know this specific spot, and so on. So that information is transported to the spinal cord and up to the brain. Myelinated A fibers will send acute pain information. Unmyelinated will send diffuse pain information. So two-point discrimination is a strategy used by physicians to identify sensitivity of various parts of the skin. So it's basically off the concept of how far apart do 2 points of stimuli have to be for you to be able to identify them as to distinguish them as separate stimuli. So for example, on your tongue you have a very high density of receptors, and so you only need to have a 1.1 millimeter difference distance between 2 points of contact to be able to identify them as different points of contact. Your hand also has very high density of receptors, so you only have to be 3 to 8 millimeters apart to be able to identify distinct stimuli. On your back is where you have a lot less density of stimuli. So 36 to 75 millimeters is enough difference for you to be able to identify 2 stimuli as distinct stimuli. So that is an important concept that is discussed in chapter 2 as well. Let's wrap up. We'll do a quick recap of the main concepts of the video. So for vision, we learned about the cornea, lens, pupil, and iris, which are parts of the eye and help to protect the eye, refract light to bend it onto the retina, allow certain amounts of light to enter the eye, and so on. We learned that the optic nerve, optic chiasm, lateral geniculate nucleus, and primary visual cortex deal with transporting and processing visual information. And then in terms of photoreceptors, we learned that there are rods which detect light in the darkness and cones which detect color. Particularly, there are red, green, and blue cones. In terms of vision, we learned about the anatomy of the ear, which includes the outer, middle, and inner ear, the malleus, incus, and stapes, which are the 3 bones of the inner ear that all vibrate to cause pressure changes in the cochlea. We learned about the vestibulocochlear nerve, which sends both vestibular or balance and cochlear or hearing information to the brain. And we learned about the semicircular tubes, which focus on localization of sound as well as balance and being able to identify your orientation. We learned about Wernicke's area, which deals with speech comprehension, and Broca's area, which deals with speech production. And then finally, for touch and pain, we learned that the receptors for touch and pain, particularly pain, are called nociceptors. We learned that there are A delta myelinated fibers which process or transport pain that is very acute and sharp, and then unmyelinated C fibers which transport pain that is very diffuse and slow. And finally, we learned about 2-point discrimination, which is the process of identifying the sensitivity of a certain patch of skin depending on its location. So with all of that being said, if you have any questions moving forward or about this video, please share them in the comments and I'll be sure to respond right away. Always feel free to reach out. And in our next video, we're going to look into chapter 3, which is about movement. So thank you so much for watching and see you soon. Bye!